fertilization and implantation. It is the process of fusion of two types of gametes, spermatozoa and ovum, so as to form a diploid zygote. Fertilization occurs in human beings and the ampulla stomach part of the oviduct. Arrival of ovum is secondary oocyte state released from ovary during ovulation, roughly in the middle of the menstrual cycle due to oscillatory current. The released ovum passes into the nearby oviduct through ostium of its fabricated funnel or infundibule. Ovum reaches the ampulla stomach junction part and rests there. It takes 12 to 24 hours for the ovum to reach the site of the fertilization. Insemination is the process of the fertilization of sperm in the genital tract of the female during copulation or coitus. The inseminated material is actually a biscuit mixture called semen. The quantity of the semen is 3 to 4 ml. It contains 80 to 100 million spermatozoa over ml. After passing into vagina, semen is converted into a coagulum. The same result formed after 15 to 30 minutes. The fetus sperm become motile as proper pH for sperm activity is provided by the ingredients of semen. Motility of sperm. Sperm can swim in a liquid medium as provided by secretion generated. The rate is 1.5 to 3.0 mm per minute. Subject develop special strings of the mucus for guiding the sperm. Prostaglandin process and in semen. Oxytocin, pituitary hormone released during orgasm of female, bring about movements of genital tract of the female, which results in the some of the sperm reaching the ampulla within minutes. Normally it takes 5 to 6 hours for the sperm to reach the site of fertilization. They can remain active for more than 24 hours. Capacitization of sperm, it is a phenomenon of sperm activation by which the sperm develop the ability to fertilize over the factor present over the sperm and in a semen which do not allow intermediate capacitation are called decapacitation factor. Capacitation developed in the female genital tract by the following processes: the lesion of inhibitory factor present in the semen, passing of the cholesterol vesicle covering the sperm, and removal of membrane cholesterol present of the this became the membrane covering entry of the Ca2 plus into sperm. This changes movement of sperm cell from lower undulating motion to rapid viscous motion, increasing permeability of the weakened acrosomal covering by Ca2 plus fusion of gametes. A sperm which has reached the ampulla stomach junction part of the oviduct comes in contact with the egg. The various events which occur during the fusion of gametes are responsible for acrosomal reaction. In contact with the surface of the egg covering the acrosome releases in content hydrolytic enzyme also called sperm lysis. It is known as the acrosomal reaction. Hyaluronidase dissolve the semantic substance of corona cell, resulting in their separation. Corona penetrating enzyme bring about degeneration of obstructing corona cells as the sperm cell reaches the zona. Enzyme bring about degeneration of obstructing corona cell as the sperm reaches the zona. The compatibility reaction Rayleigh 1919, which results in emptying of acrosome and degeneration of zona pellucida by accretion or zona lysin. Compatibility reaction also stimulate development of an outgrowth by the oocyte called fertilization or cone of precision. The compatibility reaction determined by special protein present of the zona pellucida and ulema and sperm surface. They are collectively by special protein presence of the zona pellucida and ulema and sperm surface. They are collectively called fertilization in case of egg and anti-fertilization in case of sperm. The receptor proteins of zona pellucida have identified JP1, JP2 and JP3 and optimum pH Mg2 plus Ca2 plus real sperm that comes in contact with the fertilization cone. It triggers a weak depolarization of membrane and Ca2 plus wave inside the egg. Sperm and egg membrane dissolved in the region of the contact components of head, nucleus, neck and middle piece of sperm enter the cytoplasm of the egg. Tail is left a cortical reaction. Ca plus 2 wave causes extrusion of cortical granule out of the egg. Juelas 1980 they bring about modification of plasma membrane and change the same into fertilization membrane or activation. Calix Anderson 1968 fertilization membrane does not allow entry of secondary sperm. Zona reaction chemical extracted from egg act on zona pollution and destroy the sperm receptor. Wasserman 1990 preventing attachment to any other sperm. The phenomenon by which a single sperm fertilization egg is called monosperm fertilization, where more than one sperm or polysperm is prevented by the cortical reaction, zona reaction, activation of ovum. It requires PCL zeta protein from sperm prior to fertilization. Egg is the secondary oocyte stage in which meiosis 2 has started but arrested metaphase 2 stage due to the development of MPF or metaphase promoting factor. The factor is removed anaphase promoting complex develops, which results in the completion of meiosis II and extrusion of the second polar body. This converts the oocyte into ovum. Its nucleus is now called female pronucleus karyogamy. It is the final stage of fertilization. The sperm nucleus functions as the male and female pronucleus approach each other. Their nuclear envelope breakdown. A spindle is formed. The paternal and maternal chromosome mix up, resulting in the formation of diploid set. It covers the ovum into diploid zygote, which can undergo immediate cleavage. It provides a stimulus for the completion of meiosis II. The diploid chromosome number of the species is restored. It is 46 in human. It determines the sex of the baby. 22 plus X, 22 plus X, 22 plus X, depending upon the chromosome complement of the fusing spore. Karyogamy activate the fertilized egg to undergo cleavage. There is beginning of characteristics of two parents. A lot of variation appear due to chance separation of chromosome, crossing over, and chances mixing of chromosome. It brings sperm centriole into the fertilized egg. Fertilization increases the metabolic activity of the egg. It brings about cortical and zonal reaction, which change the plasma membrane as well as gena palissuda chemically, so as to prevent entry of another sperm. Cleavage segmentation. The early repeated and retrieving mitotic division of the fertilized egg characterized by absence of growth of the outer cells are collectively called cleavage. The size of the young embryo similar to that of egg. The reduced size of cleavage cell is meant for bringing surface volume and nucleocytoplasmic ratio to the levels where metabolic efficiency become maximum. Cleavage is accompanied by rapid DNA replication, increased O2 consumption. The cells formed as the result of cleavage are blastomeres. The division which completely divide the fertilized egg into the cells of blastomere are called holoblastic. Cleavage and zygote descent begin soon after karyogamy, when the chromosome of the male and female pronuclei mingle together over the spindle called cleavage spindle. The first cleavage occurs along primary axis or axis of animal vegetal port. It takes about 30 hours. The first cleavage divides the egg into two blastomeres. One is slightly larger than the other. Therefore, the first division is holoblastic but slightly unequal. The difference in size makes the subsequent division asynchronous. The second division at least right angles to the first. It is completed earlier in one of the two blastomeres. Therefore, a transient three-cell stage is produced. 20 to 30 hours are required for completion of second cleavage. Subsequent division occurred in different phases. The third cleavage division is completed in about 12 hours. Morula stage cleavage division produces a solid ball of cells called morula. It has 8 to 16 cells, occasionally 32 cells. Morula is covered on the outside by zona palisade. It undergoes compaction. The outer and peripheral cells are smaller, flatter, or with tight junction. The inner cell mass contains slightly larger round cell with gap junction. Morula slowly descends into uterus in 4 to 6 days. The descent is caused by feeble fluid current produced by secretory and ciliary activity of mucoidal epithelium. During this period, corona radiata detaches. Importance of cleavage. It converts unicellular zygote into multicellular embryo. Rapid cell division, as the outer cells are not required to grow before the next division. Rapid metabolic activity. Reduction in cell size to attain optimum nucleocytoplasmic plasma ratio. Non-development of cell junction. This allows the cells to rearrange. Blastulation or blastocyst formation. Endometrium secretes a nutrient fluid called uterine milk. Its mucosal cells also enlarge with stored nutrients. As the morula enter uterine, it offers an enriched supply of nutrients. Therefore, there is sponge in growth. Outer peripheral cells enlarge and flatten further. They form trophoblast or trophoectoderm. The inner cell mass forms embryo. It contains stem cell which can form all types of tissue and organ. Trophoblast secretes a fluid into the interior. It creates a cavity called blastocell. The inner cell mass now chromes lie on side as embryonal. With the formation of blastocell, morula is converted into blastula, which is called blastocyst. GK blastogerm cystos bladder in mammals because of different nature of sperm layer and eccentric inner cell mass. Up till now, blastocyst has been covered by gena palissuda. However, the size of blastocyst soon becomes almost 3 times the size of the morula. Due to the presence of the growing blastocyst, a slit is produced in gena palissuda. Blastocyst may also corrode gena palissuda by tripsin like Egypt. The growing blastocysts come out of slit of gender pellucidum in the form of letter 8. At times, it gets broken down into two parts, which give rise to identical twins. Healthy separation produces conjoined twins, siamese twins. Sometimes twins are born due to fertilization of more than one egg. They are called fraternal twins, dizygotic twins. Trophoblast cells in contact with the embryonal lobe are called cells of Robert. Area of embryonal lobe could represent animal pole. The opposite side is ab-embryonal pole. Soon embryonal lobe shows rearrangement to form embryonal. These cells of trophoblast layer divide periclinically and this makes trophoblast two-layered: outer synthotrophoblast and inner cytotrophoblast. The two-layer later form chorion, amnion, and foetal type of placenta. Implantation L into placenta. It is a melting of blastocysts over endometrium of uterus. Implantation begins about 7th day after fertilization. However, it takes about 3 days for the process to be completed. Blastocyst comes in contact with endometrium in the region of embryonal lobe of embryonal disc. It adheres to the same adherence of blastocysts to endometrium, stimulate the uterine cells to undergo rapid division and partially cover the same. It results in implantation. Implantation leads to pregnancy.